Hello, mabuhay! We are the Group 2 and we will discuss about DNA replication. DNA is a molecule made up of two strands twisted around each other, but in a double helix shape. Each strand is made up of a sequence of 4 chemical bases represented by the letters A, C, G, and T. The two strands are complementary. This means wherever there is a T in one strand, there will be an A in the opposite strand. And wherever there is a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is separate the two strands. This unzipping is done by an enzyme called helicase, and the results of the formation of a replication fork. The separated strand is provided the template creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called the primer. This marks the starting point for the construction for the new strands of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously. The DNA polymerase adding bases one by one to 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore make this strand in a series of small chunks called a Okazaki fragment. Each fragment is started with RNA primer. DNA polymerase adds a short row of DNA bases to a 5' to a 3' direction. The next primer is added further down the lagging strand. Another Okazaki fragment is made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from strands of DNA. Another DNA polymerase enzyme fills in the gaps that are left behind another fragment. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old conserved strand and one new one. That's all.